Welcome students, I'm Dr. Gandhi here again, and today I'm going to teach you the process of photosynthesis and the opening and closing of stomata, both very important from exam point of view. The process of photosynthesis occurs in two phases, light phase and dark phase. To understand these two phases, we will have to understand the diagram of a cell organelle called chloroplast. So please follow me onto the board. We'll begin with the diagram of chloroplast and then gradually go on to the process. The process is very easy, don't worry. Okay? You have to only know how to go about studying the process. If you know how to go about studying the process, then it stays with you. Please don't try to rote learn. If there's any difficulty, you're most welcome to call me up. And also the opening and closing of stomata, very important. With that, we begin with the study of the process of photosynthesis, which occurs in a cell organelle known as chloroplast. Please follow me on to the board. So you can see here, I have drawn the diagram of a chloroplast. When you draw the diagram, pay attention, the shape of the diagram is very important. It is nearly watermelon shape. So you can see, I have tried to draw the shape of the chloroplast in the form of a watermelon, bulging on the surfaces, tapered on the sides. It has double membrane. If you look inside, there is a pile of flattened sacs. Remember, this pile of flattened sacs is known as thylakoids. In the walls of the thylakoid. So how do I know that in these piles what is present? So I take a cut section. So now I have taken the cut section, so I can see the inside. When I see the inside, I see under electron microscope, I will see chlorophyll. So in the walls of the thylakoid, I am seeing chlorophyll. This pile of flattened sacs called thylakoids, which includes the chlorophyll, is the first part of our study known as the granum singular, grana plural. Next, now I am going to erase these green ones because you cannot see them. Normally, our school academics only give us access to a light microscope. So I am now only going to draw some smudged appearance. But you know from my previous explanation that is chlorophyll. But you are not supposed to label it because you can't see chlorophyll under light microscope. You can only see it under electron microscope. The second part is the ground substance. The ground substance where the grana is present. The ground substance is known as stroma. So the two parts then of the chloroplast from our study are a grana, which is a pile of flattened sacs called thylakoids containing chlorophyll, and the ground substance known as stroma. In the grana occurs the phase called light phase. In the stroma occurs the phase called dark phase. So now that takes us to before I forget, the piles are connected by channels called frets of no significance for you in the exams, but you have to label it. We begin with the process of photosynthesis. The light phase occurs in the grana. The dark phase occurs in the stroma. Why is the light phase so called? Because light is required. Why is the dark phase so called? Then light is not required. Then what is required? Pay attention. Because light is not required, you call the light phase a light dependent phase. Light dependent phase. Now, at the end of light phase, pay attention again. At the end of light phase, we will get two products, X and Y. These products will be then transferred to the dark phase. So in the dark phase, you do not require light. What do you require? X and Y, which I'm going to teach you very shortly what they are. And once the X and Y are produced at the end of light dependent phase or the light phase, these products are transferred to the dark phase, therefore called light independent. They are not dependent on light, they are dependent on the end products of light phase. which combine with carbon dioxide to produce glucose. So therefore, pay attention, please do not make the mistake of saying light phase occurs during the day and dark phase occurs at night. Both occur during daytime. Both the processes of photosynthesis, the light as well as dark, occur only when the sunlight is present, that is during the daytime. But why is the dark phase so called? Because light is not required. The end products of light phase are required. That's the only reason why. So today now we are going to see what is this X and Y. I repeat again. So the end products of light phase X and Y are transferred to the dark phase, which they combine. The raw material in light phase is water. The raw material in dark phase is carbon dioxide. So again I repeat, when the light falls during the daytime, light phase occurs, which is light dependent. At the end of light phase, you will get two products, X and Y. They are transferred to the stroma, where the light independent phase occurs, meaning thereby that the products of light phase are required, not sunlight, although everything is occurring during the daytime. And when the two products combine with carbon dioxide, we will get glucose. The whole idea is to understand what is this X and Y. Once you can get that, then your study becomes very simple. So please remember to practice the diagram of chloroplast. So now we begin with the process to know what are these two end products of the light phase, the X and the Y. We begin here. Where does the light phase occur in the grana? Where does the dark phase occur in the stroma? Both occur during daytime. One requires light, therefore called light dependent. One requires the end products of light phase, therefore called light independent. Here the raw material is water. Here the raw material is carbon dioxide. We begin with the light phase. As soon as, pay attention, as soon as light falls on the grana, the first step that occurs is activation of chlorophyll. So the chlorophyll present in the grana gets activated and releases electrons. They are energy rich electrons. Only for enumeration. The most important step being the second one. In the second one, what happens? The water, which is the raw material, now splits. And when it splits into hydrogen ion and hydroxyl ion, which then becomes oxygen and water. We studied in the equation of photosynthesis, water is recycled, oxygen is released. So I can conveniently take away the hydroxyl ions because you can see here 2H2, 2OH form 2H2O and O2. So now that is not going to remain. So if I split water, it gives hydrogen and hydroxyl ion, which becomes oxygen and water. Oxygen gets recycled, water gets recycled and oxygen is released. What is this process known as? Splitting of water to hydrogen with the release of oxygen called. Light is used, therefore called photo. To split is called lysis. Photolysis of water. So I begin again. As soon as light falls on the chloroplast or the green parts of a plant, activation of chlorophyll occurs to release electron in a phase called light phase, occurring in the grana of chloroplast. The second one is photolysis of water, where water is split to form hydrogen ion and hydroxyl ion. The hydroxyl ion becomes water and oxygen. Therefore, I take it off and say that the splitting of water produces hydrogen ions and oxygen. Because light is required to split the water, the process is called photolysis of water. Next, the hydrogen ion now attaches to an enzyme known as NADP. So this the hydrogen ion gets attached and a compound is formed. The compound is known as NADPH. This is the first compound which I was talking about, X. I will tell you what is the expansion of NADP and write in the end. Nicotinamide adenine dinucleotide phosphate. Nicotinamide adenine dinucleotide phosphate, to which when you add hydrogen, it becomes reduced NADPH. The first compound we have already got. So now we go to the third one. The third one. Look carefully. When energy is used, ATP becomes ADP. Now, for any activity, including photosynthesis, we require energy in the form of ATP. But how to get the ATP? The only process we knew was respiration. And in respiration, glucose is broken down to release energy in the form of ATP. But in photosynthesis, we cannot break glucose. We have to produce glucose. Then how to get the ATP? It's very simple. To the ADP, let us say, attach an inorganic phosphate. Obviously, I will get ATP. How does this occur? Again, for any reaction, you require energy, and that comes from light. Therefore, I am writing the third one. Photo, light is required to join, relate the two, photo, phosphate to ADP, phosphorylation or relation. So this then is the third step. I begin again. In the first step, the chlorophyll was activated and electrons were released. But I wanted you to know what is this X and Y. So the first compound was NADPH. How did we get it? When the light splits water, photolysis of water. Water is split into hydrogen ions with the release of oxygen. Therefore, oxygen in photosynthesis comes from water. Next, I have already got NADPH. Now I require energy. For any reaction, we require energy. But as I told you, energy is obtained by respiration. But in respiration, glucose is broken down. But I can't break glucose because in photosynthesis I want to produce glucose. So therefore I use the energy of light to join a phosphate. So now how did the ATP get formed? To adenosine diphosphate, I added an inorganic phosphate coming from the soil as a result of active transport. And when the two join, I get ATP. The second chemical required. So I got my two chemicals. So I'm writing the two chemicals here. I got the first one. The first one is called NADPH. Nicotinamide adenine dinucleotide phosphate reduced as a result of photolysis and hydrogen combining with an enzyme. The hydrogen combining with an enzyme known as NADP. Next, for creating ATP to synthesize glucose, I combined inorganic phosphate to ADP to form ATP, known as, with the presence of light, known as photophosphorylation. I got the second one, ATP. So these then are the end products of light phase. Now it is very easy to understand. In the dark phase, I have got carbon dioxide. I now bring the two products of light phase into dark phase. The two products being NADPH and the other product being ATP. So the energy has come. What did the energy do, or what was it required for? For transferring the hydrogen to carbon dioxide. So now with the help of energy, I am going to transfer the hydrogen to carbon dioxide, forming the basic molecular formula of glucose. So the hydrogen got transferred, and in this reaction, ATP became ADP. and we got glucose. We got the glucose, you have to just balance it. So we got glucose. As simple as that. So you can see glucose has been produced. Okay. Now, at the end, you got NADP. The NADP comes back in the light reaction to join with hydrogen, again become NADPH. The ADP comes and joins with inorganic phosphate to become ATP. Therefore, photosynthesis goes on during daytime. This then is the basic process of photosynthesis. The two very important steps are photolysis of water and photophosphorylation. If somebody were to tell me to define photolysis of water, it is the splitting of water into hydrogen ions and oxygen using energy of light in the grana of chloroplast. Define photophosphorylation. It is the formation of ATP by combining inorganic phosphate to ADP using energy of light in the grana. Grana means presence of chlorophyll in the grana of light. The two end products of light phase were now taken to the dark phase, and with the help of energy produced by light, hydrogen gets transferred and glucose is produced, the ATP becomes ADP. So what is the concept of photosynthesis? Pay attention. Light energy becoming chemical energy. Because if there is no chemical energy, the reaction would not occur. So in photosynthesis, light energy is converted to chemical energy. And what is that chemical energy? We all know. ATP, the basic concept of photosynthesis. So that takes care of light and dark phase. The enzyme required in light phase to accept hydrogen is called NADP. Now, the carbon dioxide also requires to be present in the chloroplast till the end products do not reach. So there is a carbon dioxide acceptor in the stroma or the dark phase in the stroma, and that is known as RUBP, ribulose bisphosphate. So name the carbon dioxide acceptor in dark phase, RUBP. Name the hydrogen acceptor in the light phase, NADP. Okay, so that takes care of the process of photosynthesis. I begin again. The process of photosynthesis occurs in two parts, light phase and dark phase. Light phase occurs in the grana, dark phase occurs in the stroma. So called because light is required, dependent on light. Why is the dark phase so called? Because the end products of light phase, NADPH and ATP, are required. Therefore, it is light independent. Although light is present, it does not require light. Therefore, light independent. So remember, photosynthesis, both phases occur during daytime. The two important reactions in the light phase are photolysis of water and photophosphorylation. The reaction in the dark phase is known as reduction of carbon dioxide to any substance. When you add hydrogen, it gets reduced, therefore called reduction. At the end, we have got glucose. Several molecules of glucose, mind it, many molecules of glucose join to form one molecule of starch, a process called polymerization. So now, poly means many, mers means part, polymerization. By a process called polymerization, glucose is stored as starch. So that takes care. Again, an important enzyme which accepts hydrogen in the light reaction is known as NADP. Carbon dioxide acceptor in dark reaction is known as RUBP. define photolysis of water. It is the splitting of water into hydrogen ion with release of oxygen. Oxygen in photosynthesis comes from water. Photophosphorylation, no, sorry, I'm defining photolysis of water. It is the splitting of water into hydrogen ion and release of oxygen using energy of light in the presence of chlorophyll or in the grana of chloroplast. Explain the term photophosphorylation. It is the formation of ATP by combining inorganic phosphate to ADP in the presence of light using energy of light in the presence of chlorophyll or in the grana of chloroplast. What is the energy conversion in photosynthesis? Light energy to chemical energy known as ATP. What are the end products of light phase? NADPH and ATP. What is the end product of dark phase? Glucose. What is polymerization? Many molecules of glucose become one molecule of starch. So that takes care of the process of photosynthesis. I'm now going to write down the full form of NADP and RUBP. NADP, nicotine. Don't write E, please. Nicotinamide, adenine, dinucleotide, phosphate. What is the full form of RUBP? Ribulose, bisphosphate. What is NADP acceptor of? Hydrogen ions in the light phase. What is RUBP? Carbon dioxide acceptor in the dark phase. We now go to some other terms which are associated with light phase and dark phase. The first one then, you can see at the end of light phase, two chemicals are produced, NADPH and ATP, therefore called photochemical reaction. So give another term for light phase, photochemical reaction. The next one, here, you come here, here you can see glucose is produced, therefore called biosynthetic phase. So the other term for light independent phase is biosynthetic phase. For light phase is photochemical. A scientist who worked has given his name to it, Hill, therefore known as Hill's reaction. The scientist, a colleague of Hill who worked in the biosynthetic phase is known as Calvin, therefore called Calvin's reaction. So the various names then are light phase, light dependent phase, photochemical phase, Hill's reaction. Dark phase, light independent phase, biosynthetic phase, Calvin's reaction. Okay, so that takes care of the study of photosynthesis. Let me erase this. I will now go back to the cut section of a dicot leaf which we had studied in transpiration. But I will refresh your memory and take it from here, take up the explanation towards opening and closing of stomata. Can I have the chart, please? Thank you, Swati. I'm sure the students will bless you for being such a help. Swati is here to help me with my live video recording. I'm very, very grateful to you. God bless you. Next. So now, you can see this then is the cut section of a dicot leaf we studied. This is the palisade parenchyma, spongy, and these are guard cells. The palisade parenchyma is on the upper surface of a dicot leaf. Remember, in a monocot leaf, there are only spongy mesophils. In a dicot leaf, you have palisade and spongy mesophyll. In a dicot leaf, where is the chlorophyll present? The chlorophyll is present only in three locations. One, palisade mesophyll. Two, spongy mesophyll. And third, guard cells. These are the three locations where we have chlorophyll. And this understanding that guard cells has chlorophyll will help us to understand the opening and closing of stomata. So this then is the cut section of a dicot leaf. When I resume personal classes and when the school reopens, obviously, at that time, if not understood, I will take care of all the chapters. I will go through them quickly so that we don't miss out on any understanding whatsoever. So please relax, try to understand the basics of a chapter. If you do not understand, I have told you, you are most welcome to put a WhatsApp on my personal mobile and I will help you. So now, with this idea that guard cells contain chlorophyll, and in a dicot leaf, gave reason it is a dicot leaf, palisade and spongy. Then in a monocot, what will you only find? Spongy. So what is this then? The gut section of a dicot leaf, palisade and spongy. Okay, and now we'll begin from here. Opening and closing of stomata. You can see with the help of guard cells. So now I begin with the study of opening and closing of stomata with the help of guard cells. It is a very, very beautiful phenomenon. Very, very beautiful. If you understand how nature is so brilliant in constructing certain things, you will be amazed. And the more you are amazed, you will start liking the subject. The problem is we do not get enough joy when we study. So I'm trying to help you to get joy so that a subject not only becomes easy, but it amazes you to study further. So I'm going to now draw the guard cells surrounding the stoma. The beauty of the guard cells, as I draw, you will realize. Therefore, I request most of my students, or all of my students, please draw. When you draw, the detailing unfolds, and when the detailing unfolds, the explanation unfolds, and when the explanation unfolds, the subject becomes easy. Then it's very easy to read the textbook. So look at the way I'm drawing the guard cell. Okay, the guard cell is bean shaped or kidney shaped. Look here. This is the shape of the guard cell. I'm going to draw the other side, but I'm going to focus only on one guard cell at the moment. A guard cell surrounding the stoma. Please make it a habit or a practice to draw. When you draw, it is a very important rehearsal for your exams. If you draw, you know the subject. Next, the guard cell has got a wall. Look here. This is the outer wall. I'm drawing the outer wall thick. Look here. The outer wall is thick. The inner wall is thin. So the first characteristic then of the guard cell to help in opening and closing the stomata is that the inside wall is thick. So I'm drawing inner wall thick, outer wall thin. This then is the cell membrane. This then is the cell membrane. This then is the nucleus. Okay, next. What is the second characteristic feature of guard cell which makes it possible to open and close the stomata? Presence of chloroplast. The chloroplast in its grana contains chlorophyll. So all these are chloroplasts. So you can now see a guard cell has two characteristics because of which it can open and close the stoma. Inner, because the stoma is here. Inner, this is the inner, this is where the stoma is going to be. Inner thin wall, outer thick wall, and presence of chloroplast. So I'm now going to draw on the other side. So now I will draw on the other side. You can see again I'm drawing the inner wall thick. The inner wall is thick because this is the stoma. Singular stoma, plural stomata. Plural is stomata. You can see the inner wall is thick. The outer wall is thin. This then is the cell membrane. This then is the nucleus. And this then is the presence of chloroplast. So now the two features which are going to make it possible for a guard cell to open and close the stomata are inner thick wall, outer thin wall, presence of chloroplast. At the moment you have a diagram of an open stoma. Next, where are the stomata present? They are present in the epidermis of a leaf. So I am now drawing the epidermis. These then are the epidermal cells surrounding the stomata. I am only drawing two, but they are all around. These then are the epidermal cells. You are supposed to draw all around. I am just drawing on one side. This then is the epidermal cells. The epidermal cells along with guard cells are going to teach me a process called opening and closing of stomata. In your laboratory, when processes are explained, they are explained by means of apparatus. The epidermal cell and the guard cells are going to teach me a process called opening and closing of stomata. Therefore, this diagram is called stomatal apparatus. Identify the diagram showing epidermal cell and guard cells. Stomatal apparatus. We now go and see why the guard cells open during daytime. Can you see here? Okay, thank you. Daytime. So pay attention, now we are in the guard cells. So look here, in the guard cells, because of presence of chloroplast and in the daytime sunlight occurs a process called light phase. Occurs a process called, photosynthesis. And in photosynthesis is produced glucose. So now glucose is produced. Look here, this is glucose which is produced. This is glucose during the daytime produced in the guard cells. Now, when the glucose is produced in the guard cells, the solution in the guard cell becomes hypertonic. Compared to, whenever you use the word tonic, you have to use the word compared to. The epidermal cells, the solution of the cell sap in the epidermal cells now remains hypotonic. So these are hypotonic. This is hypertonic. So you know water goes in osmosis from hypotonic to hypertonic solution. So water during daytime enters the guard cells. Now the water has entered the guard cells. Obviously, when water enters the guard cell, pay attention, during daytime, the guard cells now become turgid, firm. So now the guard cells have become firm. Now pay attention. If you have known that if you have a balloon, this will give you an idea. If you have a balloon in which you push in air, we have commonly observed that the part of the balloon which is fragile bulges out because of the pressure of the air. The weak part or the thin part bulges out. So if you see that water has entered, air enters, thin part bulges out. So if water enters a guard cell, the thin part bulges out from both sides. And look here, I'm drawing. When they bulge, they open the stomata between them. So now that is the reason when the guard cells become turgid, pay attention, the guard cells become turgid and bulge outwards. And when they bulge outwards, they split open the stoma. That is the reason why stomata open during the day. I'm repeating it because many students have put a WhatsApp, sir, we did not understand opening and closing of stomata. I begin again. Two characteristics of a guard cell making it possible. Inner possible to open and close the stomata. The first one, inner wall thick, outer wall thin, and presence of chloroplast. We begin during the daytime. In the guard cell occurs a process called photosynthesis, because of which glucose makes the cell sap in the guard cell hypertonic compared to the epidermal cells which are hypotonic. In osmosis, with respect to tonicity, water goes from a hypotonic to a hypertonic solution. So now water enters the guard cell. When water enters the guard cells become turgid, and because the inner wall is thick and the outer wall is thin, the weaker part bulges out, and therefore the guard cells bulge out. And when they bulge, the stomata opens. Opposite happens at the time when photosynthesis does not occur. So at night time we will see now what happens. So now we change this into our understanding into night time. So we change all this understanding into night time. So we begin at night. A process called translocation occurs. What is translocation? Food. So in this case, you can see the glucose which was present in the cell during the daytime, now the glucose moves, translocates to the epidermal cells. So now the glucose in the epidermal cell causes the solution in the epidermal cells to become hypertonic. So the cell sap in the epidermal cell becomes hypotonic because the sugar has moved away from the guard cells. Comparatively, the cell sap in the guard cell becomes hypotonic. And you know now, water goes from hypo to hyper. So water leaves the cell. So at night time, water moves out of guard cells, and the opposite of turgid happens. What do the guard cells become? Flaccid. And when they become flaccid, like when you remove air, the balloon becomes flaccid. So now at night what happens? The guard cells close. So now showing you the diagram of a closed guard cell. In the exam you will get to draw. So now the guard cells have closed the stoma, or the singular being stoma. So now, look here. I want you to pay attention to what I'm doing. So my diagram showing a closed stoma. Because you will get this to draw in the exam, I want you to highlight, please do not draw like this. The reason being, if you show a closed stoma like this, you will not get marks in the exam. Why? Because we forgot at night occurs a process called respiration. And in respiration, which occurs 24/7, oxygen has to enter. If you show a guard cell closed like this, then how will the oxygen enter? How will the carbon dioxide come out? Therefore, you have to draw a small slit inside. Pay attention. This then is the diagram of a closed stomata. You can see a slight opening so that oxygen can enter for respiration and carbon dioxide can leave out. This then is known as the sugar theory of opening and closing of the stomata. The other theory, which is widely accepted, is that during the day, because of sorry, in the first one, ATP is produced because of photosynthesis. What does the ATP do? It pulls in from the epidermal cells, potassium ions enter the guard cells, making them turgid, or making the solution, because of potassium ions, they become turgid, and when they become turgid, or the solutions become hypertonic compared to hypotonic. Previously, sugar made the solution hypertonic. Now it is the potassium ions, because of which what does the guard cell become? Turgid. And why does the stomata open? Because the inner wall is thick, outer wall is thin, the cells bulge outside, and the guard cells open. At night, there is no photosynthesis, so there is no ATP. Potassium comes back to the epidermal cell, again making this solution hypotonic and hyper. Water leaves the cell. So at night, again, the same thing. In the flaccid and the stomata close. Please do not close it. So what is the difference between sugar and potassium theory? Sugar glucose made this hypertonic. And in potassium theory, because of ATP, potassium entered and made it hypertonic. That is the only difference. So now we studied two theories, the sugar theory and the potassium theory, to know the reason for opening and closing of stomata. But in the exam, they will ask you, tell me one reason, give one reason for opening and closing of stomata. I am not interested in theories. I want to know what is the basic reason. It can be any theory, whether it is a sugar theory, a potassium ion theory, irrespective. Give one reason for opening and closing of stomata. Okay, that one reason is water entering and leaving the guard cells. So when I send you markings, please make a note here. When water enters a guard cell due to any theory, the stomata open. And when water leaves the guard cells, the stomata close. So this is the main reason for opening and closing of stomata. I'm not talking about the theory, I'm talking about the practicality of the opening and closing of stomata. In the stomatal apparatus, the water enters the guard cell, stomata open. And when the water leaves the guard cells, the stomata close. Identify the diagram, stomatal apparatus. So that takes care of the chapter photosynthesis. In the first part, I taught you de-starching and starch test. The main conceptual words for the experiments required to study carbon dioxide, sunlight, and chlorophyll required for photosynthesis. I may have used the word external, but remember chlorophyll is an internal factor for photosynthesis. In live recording, you sometimes tend to miss. So I'm now correcting myself. Okay, so the external factors are carbon dioxide and sunlight, the internal factor being chlorophyll. Next, today we studied the process of photosynthesis, light phase, dark phase, diagram of chloroplast, and the explanation. And we also studied the opening and closing of stomata, stomatal apparatus. If you have any difficulty, you are most welcome to call me up on my number. Stay home, stay safe, stay blessed. God bless you.